Hello everyone, welcome back to our discussion on mutations. So we in the last section we have completed our discussion on chromosome mutations, where we discussed about the change in number of chromosome and also the change in structure of chromosomes. And in this session we are going to discuss about gene mutation or point mutation. These are the changes in the base sequence of the gene, and that will result in an abnormal protein to be formed, or that may affect the synthesis of the protein, etc. So, depending upon where the mutation occurs. So, gene mutation or point mutation can either occur in the gene region. So, in the gene region means if you take the DNA, if I'm taking this as a double-stranded DNA, okay. So consider this as the double-stranded DNA with the base pairs. Okay. So this is the double-stranded DNA, and so I'm not drawing every base pair. So consider this as a very long DNA, and for say from here to here you have a gene. For say this is an insulin gene. Okay. So this is the gene region. Okay, so this is the gene region. And if mutation occurs in any base in this particular region, what happens is this will affect the production of insulin, or a protein will be formed, but that might not be insulin, etc. So that is how a gene mutation or a mutation in the base is affecting the function of that particular gene. So mutation can be so this is a gene region, where this is a gene region. It can happen in the gene region, or there are the regions which is not included in the gene, or that is not part of the protein, but these are the regions which are usually involved in regulating this particular gene. Regulating in the sense, for this gene to produce an mRNA, there should be a place for binding of the RNA polymerase. There should be many regulatory sites. So what happens is mutations can also, gene mutation or point mutation can also occur at this regulatory sites also. If this happens in the regulatory side, what happens is that either this particular, usually if it happens in the regulatory site, for example, this is a site where your RNA polymerase, which results in the, or which is an enzyme which is responsible for the production of an mRNA from this DNA. So this is the mRNA, single stranded mRNA. So what happens is, if this is affected, the RNA polymerase, the enzyme will not bind here. As a result, the mRNA will not be formed. But if it is in the gene region, this regulatory regions are normal, so RNA polymerase will bind. It will produce a protein, but this will not be like normal insulin. It will be either a crippled protein, it will be a longer protein, or it will not be insulin, it will be some other protein, etc. So these are the consequences of gene mutation. So gene mutation or point mutation can happen either in the gene region or the regulatory region of the DNA. So coming to gene mutation or point mutation, it refers to the alteration of a single base or single nitrogen base in the DNA, or small number of adjacent bases. It can be single or more than two, etc. So there are two main types of point mutations in DNA. Uh, they are base substitutions and base insertions or deletions. So these are the two main types, either base substitution in the sense, in some cases adenine will be replaced by guanine, or if it is guanine in the normal one, will be replaced by thymine, etc. So these are called as base substitution. Base insertion is that some bases, extra bases will be inserted, or base deletion in the sense some will be deleted. So these are the different types. And base substitution mutations are in which one base is replaced by other, and usually substitution is divided into two. One is called as transition and the other one is called as transversion. So these are the two different types of substitution or gene substitution mechanisms. First we will see what is a transition. So it is also a substitution. if a purine is replaced by a purine, that is a replacement of base by another one which is of the same chemical category. That is, we only have two different types of two chemical types of nitrogen bases. One type is purine and the other one is pyrimidine. And the two purines are adenine and guanine, and the two pyrimidines are cytosine and thymine. So what happens is, if a purine is replaced by a purine, that is, if adenine is replaced by guanine and guanine is replaced by adenine, so they belong, because they both belong to the same chemical family of purines. Such a type of substitution will be called as transition. So that same is applicable for pyrimidine. If cytosine is replaced by thymine and thymine is replaced by cytosine, vice versa, such substitutions are called as transitions. Next, coming to transversion, which is opposite of transition, that is the replacement of a base of one chemical nature category is replaced by a chemical or a base with another chemical nature. That is, when a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine, uh, that type of substitution is called as transversion. So either pyrimidine is replaced, in both cases can happen. So you can see here, when a cytosine is replaced by adenine. So this is a pyrimidine and this is a purine. So this replacement is called as a transition, C to G, T to A, whatever it is. So a pyrimidine is replaced by a purine, or a purine being replaced by a pyrimidine, because they are of opposite chemical nature. Such substitutions are referred to as transitions. So now we are discussing gene mutation. In that gene mutation, we said that there are mainly two classifications of gene mutation, broad classifications. One is base substitution and the other one is base insertion or deletion. So base substitution, in the sense that one nitrogen base will be replaced by the other. So in that itself, depending upon the chemical nature of the bases, nitrogen bases in DNA, you have two chemical categories. One is purine and the other one is pyrimidine. So on the basis of the chemical nature, substitution can be of two types. Transition, where uh, one base is replaced by the other, which is of the same chemical family or chemical category, which is a purine to purine replacement or a substitution, or a pyrimidine to pyrimidine substitution. If the opposite happens, when a purine is replaced by pyrimidine or a pyrimidine is replaced by a purine, what happens is, or that particular process is called as transversion. So these are the two categories. So now we'll see the different types of or the molecular consequences of point mutations in the coding region. We'll see transition, transversion, and the different types of transition, transversion, base insertion, base deletion, etc. Okay, so in this particular case, we'll be discussing all the different types of gene mutation, that is base substitution, base insertion, and base deletion. So this particular table is a representation of all the type of gene mutations that we are going to, or that we are discussing in this particular session. So here you can see a wild type. That means the normal one. Actually, this they have. We are taking here the DNA. Actually, this DNA will be converted to an mRNA, and it is this mRNA that is going to give you this particular thing, particular protein sequence. Okay, so here we have taken it as a DNA. That's why there is a thymine which is written here. When it is transcribed, it will be transcribed into an mRNA. It will be converted to the complementary strand. So here we are taking this as a DNA strand, because usually this mutations can happen on the usually it happens on the DNA. Okay, it happens on the DNA, and that is will be reflected to the mRNA, and that will be reflected to the protein. So that's why we are taking the DNA sequence for our discussion here. So here it is a normal DNA sequence. So these are the four codons. So this three letters will code for a particular amino acid. this three letter will code for another. So three letters are coding for one amino acids, and this will be joined by peptide bonds to form a protein. So here you are taking a peptide with four amino acids, and the normal ones are this is threonine, this is lysine, this is arginine, and this is glycine. So this is a normal sequence. So now we are going to see what are the effect of point mutations, or what will happen when a point mutation happens in this particular region. So first we are going to discuss the major one, which is base substitution. So this is base substitution. So it can be either transition or transversion. So we'll check whether this is a transversion, or each one is a transition or transversion, when we discuss one by one. So the first one, you can see, so this is the DNA sequence, and in the third codon, what happens is that the last one, that is here, it is adenine. Adenine is replaced by cytosine. So adenine is a purine and cytosine is a pyrimidine. So this is transversion. So the first example is a transversion, and you can see that even though transversion occurred, what happened for this particular one? So this is a point mutation because there is a change in DNA sequence here. So even though there is a change in DNA sequence, it is coding for the same amino acid. Is that clear? So a point mutation occurred, but that point mutation is not reflecting in the amino acid which is being coded by this particular codon. Okay, so this is because the property of the codon, which is called as the degeneracy of the codon. That means because for some amino acid, not for all amino acid, some amino acid have more than one codon for coding that particular amino acid. And one example is arginine. Arginine can be coded by AGA. If the code is AGA, arginine will come. If the code is AGC also, arginine will come. So this point mutation is a mutation because there is a change in the DNA sequence, but that does not reflect in the particular protein. Protein is a normal one. You can see this is a normal sequence. This is also the normal sequence. So this is a transversion. Okay, this altered codon is specifying the same amino acid, and such mutations are called as synonymous. Synonymous means there is a mutation, but that mutation is giving you the same meaning. That is why it is called as synonymous. So this is the first type of substitution, where even though a mutation occurs, that does not change the protein that is being produced. Okay, the reason for that is due to the degeneracy of the codon. Degeneracy in the sense, for some amino acid, there is more than one codon to code for that amino acid. So some amino acid are having six codons, some are having four, some are having three, some have two, and there are also some amino acid which has only one code. For such amino acid which is having only one code, a single mutation or a single mutation will change that particular the code for that particular amino acid. Okay, the second type of substitution is called as Missense mutation, and that Missense mutation is conservative. So first one was synonymous in the sense that it gives you the same protein sequence. So here we are going to see the second type, where you can see this guanine here is replaced in the third codon. Guanine is replaced by adenine. So both are purines, and so here this base substitution is called as transition. So this is transition. So this particular transition, what happens is, here you can see, instead of arginine, now another amino acid is being coded, and that amino acid is lysine. Okay, lysine is coded instead of arginine, and this is called as missense mutation. Missense in the sense, now the sense of the protein is lost. Here, the sense of the protein is conserved. Here, the same protein. That is why it is called as synonymous. Missense in the sense that you are getting an altered protein here, so that the sense is lost. That is why it is called as missense. And even though it is a missense mutation, it is called as conservative. It is called as conservative because both arginine and lysine belong to the same chemical family of amino acids, which is they are they belong to the category which is called as basic amino acid. both arginine and lysines are basic amino acid because their chemical nature, because there is an amino acid replacement because of a transition, and the replaced amino acid belong to the same chemical category of amino acid. Both, that means the first one was a uh, basic amino acid that is again replaced by another basic amino acid, and that is why this missense mutation, even though the protein is different, its chemical nature will be conserved. That is why it is called as conservative missense mutation and the conservative mutation. The altered codon specifies a chemically similar amino acid. Okay. Next is missense mutation, which is non-conservative. So here the same thing, and guanine is replaced by thymine, purine replaced by pyrimidine, which is transversion. So in this, what happens is this is replaced by chemically different. So arginine was, as I said, arginine was a basic amino acid which was replaced by isoleucine, which is a hydrophobic amino acid. So they are chemically different or dissimilar. That is why. So the already the protein is different. So that is missense mutation, and the mutation is non-conservative because the chemical nature is not conserved here. And another type of substitution is called as nonsense mutation. So here you can see that in the second codon, instead of adenine, a thymine is being replaced in the first codon, in the first position, not the first codon, first position. Adenine is replaced by thymine, which is transition. So in this altered codon is specifying a stop codon. As a result, after adding this particular amino acid, the process of the process of translation will be halted because it is a stop signal, a stop codon. As a result, the entire protein is the protein itself is not produced, and here you can see that the altered codon is signaling for chain termination or termination of protein synthesis, and such mutations are or such substitutions are referred to as nonsense mutation. So there are four different classes of, or four different types of uh, substitution mutations. One is synonymous, the second one is missense conservative, the third one is missense non-conservative, and the fourth one is non-conservative. So in all this three, you can see that even though there is a mutation, the number of apart from this nonsense mutation, the number of amino acid formed is the same. Okay. The altered codon is specifying the correct number. The frame is not being shifting. Okay, the reading frame is the same. That is, the reading frame is this 3 3 3 3. Here also it reads 3 3 3 and 3. There is no change in the reading frame for these three. In this one, because of the stop codon, the entire thing is gone. Okay, so next is we are going to discuss about base insertion and base deletion. Together, they are usually called as indel mutations or frame shift mutation. They're called as frame shift mutations or indels because of an addition or because of a deletion, the reading frame itself is entirely changed. Here the reading frame is not changed. Here it reads correctly 3 3 3 etc. Only that the mutant part is being read as different one or a different version. But here the entire frame or the entire reading frame will be altered because of either an addition or a deletion. So you can see here, that is the first one is base insertion or adhesion. This is a frame shift mutation or an indel. Frame shift in the sense that you can see here, a guanine is inserted in between. As a result, you can see, so this is a normal one, ACA, and other one is AAG. So instead of reading AAG or the other three, because a G is inserted, now the ribosome for adding this amino acid, it will read GAA. So the normal frame is now changed. Okay, the usual frame was reading frame was AAG. Instead of that, because this is inserted, now this will read because ribosome will is having the property to read only three letters. Constitutive three letters will be read by the ribosome. So when G is inserted, it will change the entire reading frame. So here you can see that this mutation in this particular substitution, what happens is only the substituted codon is having the problem. The other ones are the same. Other ones are unchanged. But here, because of insertion, From the point of insertion, downstream, all the amino acids are changed. That is why it is called as frame shift mutation. The same thing happens for a deletion. Here an adenine is deleted. When deletion occurs also, the entire frame, you can see from the point of deletion, that is why it is represented in red. The entire frame or the reading frame, the entire amino acid from downstream of the site of uh, deletion or addition is changed, and that is why they are called as frame shift mutation, and or also called as indel mutations. So base insertion and base deletion, addition and deletion is collectively called as frame shift mutations or indel. Okay, and this will result, both have the same consequence, that is they result in the change in the reading frame of that particular mRNA or that particular DNA. So this is about the consequences of point mutations within the gene region. So these are the definitions of synonymous, missense, and nonsense mutations. So next is the molecular consequence of point mutation in the non-coding region. So that was in the coding region. So what happens in the non-coding region? So non-coding region, as I mentioned in the beginning of the session, that non-coding region are regulatory regions. They do not code for protein. They regulate the production of mRNA. So they are not directly involved in coding the protein and may contain crucial DNA binding sites for proteins which are interspread among the sequence, etc. So at the DNA level, the binding sites include the sites for RNA polymerase and associated factors, etc. And at the RNA level, these the non-coding regions in the RNA are important for ribosome binding, then splicing of the RNA, etc. So these are not directly involved in the protein sequences or the mRNA sequence, but they regulate the processes, either transcription or translation. So the functional consequence depends on whether the mutation disrupts the binding site. So such regulatory mutations usually alter the amount of protein which is being produced. In some cases, the amount is affected, or in the other case, the entire protein production will be halted. Okay, because when the RNA polymerase cannot bind, when there is no specific splicing machinery, etc., the entire protein formation will be halted. So this is a consequence which is shown here. So here, what they have shown is, you have a wild type gene here, an mRNA being formed, a protein is being formed, and this is an experiment or a particular procedure where you analyze the RNA as well as the protein. Okay, so this is called as blotting, where in northern blotting you analyze the mRNA and in western blotting you analyze the protein. So this is a band, depending upon the size of the mRNA and protein, it will attain a specific position in the band or a specific position in this particular blotting paper. So what happens is, this is the normal the position of the mRNA, and this is the normal position of the western blotting, or normal position of the protein. So here what happens is a missense mutation. The missense mutation, there is a mutation, and you can see that uh, because of the mutation, the size of the mRNA does not change, because it is a missense, where we said that it is not addition or deletion, it is a substitution. So because of substitution of the mRNA, the size of the mRNA does not change. There is a change in the sequence, but not with the size, so that they are attaining the same site in the gel or the paper. So, but you can see that the protein is altered. Okay, there is a small change in the protein which is being formed, and this is a nonsense mutation. Because of nonsense mutation, the entire protein is being formed. It stops in between entire sequences, so that the size of the protein is very less. And as I said, here the separation is based on the size. uh, this is a lower size, so it attains a position which is very lower to the mRNA. The mRNA will be the same. Okay, mRNA does not have any change. So here this is a frame shift mutation. So here in frame shift mutation, you can see that depending upon addition or deletion, that is why the arrow is like this. Okay, if bases are added, more bases are added, the size of the protein will increase, and that will attain a position somewhere here. If more bases are lost, it will attain a position here. So the consequence of a regulatory region mutation is that there is no mRNA, no protein, and if you run this, there is no bands because there is no mRNA, no protein, because the regulatory region was affected. No mRNA was produced. No mRNA means there is no protein. So this is the consequence of uh, what happens in gene mutation, if it is happening in the gene region or if it is happening in the regulatory region of the gene. So with this, we come to the end of our discussion of different types of gene mutations. In gene mutations, we discussed about the base substitution, the four different types of base substitution, then base insertion, base deletion, which is also called as indels or frame shift mutations. So in the next session, we'll be discussing about the molecular basis of induced mutation, which is induced by chemicals, by radiations, etc. So that we'll discuss in the next session. Thanks for hearing.